We spent a great deal of time at the beginning of the year on organic chemistry, and we covered the structure and function of carbohydrates. We covered the structure and function of lipids and the structure and function of proteins, and we did it in a fair amount of detail. I left off covering nucleic acids of any kind because nucleic acids are so closely related with genetics and passing inherited information on from parents to offspring that I felt like it would be more appropriate to learn about the structure and function of those when we do our genetics unit. Okay, so for a little bit of history, which I actually think is important because this quote-unquote history is really new. Okay, the structure of DNA was just found in 1953. We're getting pretty close to the 60th anniversary of that occurrence. So, this was something that was discovered within our grandparents' time. And science and medicine have changed drastically in the past 60 years because of it. The rate of change has been much more quick than it had been previously, because once we discovered that, it was sort of like finding the keys to a treasure box. It gave us a lot of power to discover many other things. Okay? Before DNA's before DNA's structure was figured out by two gentlemen named Watson and Crick, and what they did was they really pulled scientific information together from several other scientists. One scientist named Rosalind Franklin, who was doing X-ray crystallography. It's a technique that shoots X-rays at crystallized structures of things, and then they see the scatter pattern, and what that allows them to do is sort of figure out the structure. It's kind of like figuring out that somebody is a person based on their shadow, or that a pig is a pig based on its shadow. You kind of see the shadow it casts and see what it looks like. So they used data from Rosalind Franklin, and they used data from several other scientists as well who had sort of figured out how many nitrogenous bases there were and that there were some phosphates in there. So Watson and Crick pulled together information from many other scientists to figure out the actual structure of DNA. And what they discovered was that DNA is what we call a twisted double helix. Now that's redundant. We call it a double helix, okay? But it's like a twisted ladder. And you can see that in this picture. What this picture shows, you can see the rope that I'm highlighting along the side. This is the sugar phosphate backbone, okay? And the sugar phosphate backbone is made of, in the case of DNA, the carbohydrate deoxyribose and phosphate groups, and it forms the backbone because it's the outside and it provides a lot of the structure of the molecule. And then what you see here in pink and in blue, and they look like bubbles, are the nitrogenous bases. And the order of the nitrogenous bases is what makes the genetic inherited information that's passed on from parent to offspring. Okay? This whole thing, this picture of DNA, is actually called a nucleic acid. So we're gonna look at nucleic acids and what they're made of. Nucleic acids are actually long strings of things called nucleotides, okay? And one nucleotide is made up of three basic parts. There's a phosphate group, okay? If you look up on the side cabinet, you'll see that phosphate groups, they can provide energy, for sure, but that's not their function here. The phosphate group functions here to, it makes the nucleic acid water soluble, and it can absorb hydrogen ions. There's a carbohydrate here. This is the sugar, okay? And we'll talk a little bit more about that later. And then there's a nitrogenous base. Now the word nitrogenous is because it's got so much nitrogen in there. See that? And it's a base because it can provide hydrogens, okay? So every nucleotide has this basic structure, and the only things that change are the structure or which nitrogenous base is on there, okay? All right. So this molecule, DNA, is made up of two facing chains of nucleotides. So sometimes we can call nucleic acids polynucleotides, actually. Okay, so let's take a look at each component. Nucleic acids are either DNA or RNA, and I'm pretty sure you've heard both names. And so these are the sugars that are in them. Deoxyribose is in DNA, and that's this top sugar here. Notice it's a pentose. That means it's a sugar with a 5-carbon ring. So this is carbon 1, 2, 3, 4, and there's carbon 5 up there. Okay, so deoxyribose has this structure. Ribose is the sugar that's in RNA, and let's take a look at its structure. It's a 5-carbon ring, and the difference between ribose and deoxyribose is, if you notice, deoxyribose on its second carbon here only has a hydrogen molecule, 2 hydrogen molecules actually. Ribose on its second carbon has a hydroxyl group and a hydrogen molecule. Get it? So deoxyribose doesn't have a hydroxyl group on its second carbon, whereas ribose does have a hydroxyl group on its second carbon. And that actually affects the reactivity of the molecule and what it can bond with, and it sort of changes the structure of RNA and DNA. Okay. Then there's the phosphate group. Okay. The phosphate group helps to connect the sugar backbone, so it connects the deoxyriboses or the riboses, depending on if it's DNA or RNA. It actually will also bond to the carbonyl carbon of one pentose to the carbon in position 3 of another. So let's take a look. When I talk about, here we go, the carbonyl carbon, okay, remember a carbonyl or carbonyl, C-A-R-B-O-N-Y-L, is a carbon, it's this one right here, okay, the CH2OH. Okay, so the carbonyl carbon through the phosphate group will bond with carbon 3 on another. So this one bonds to a phosphate group, bonds to this carbon here. All right, and we'll take a look at that. You can see it here. Here's the carbonyl carbon. Okay, and then there'll be another phosphate group down there. And then as well, the phosphate group will tend to ionize. So when it ionizes, it can lose a hydrogen or gain a hydrogen very easily. So it makes it water soluble and it increases its stability. It actually can act like a buffer, absorbing hydrogens or releasing hydrogens. Okay, so we've gone over the carbohydrate, the phosphate group, and now the nitrogenous base. Now there are 2 classes of nitrogenous bases. They're called either purines, and the purines are here, adenine and guanine, or pyrimidines, and the pyrimidines are here, and there are 3 types of pyrimidines. There's thymine, cytosine, or uracil. Okay, the purines, as you'll notice, are 6-carbon and nitrogen ring. Okay, so this is the 6-carbon and nitrogen ring fused to a 5-carbon and nitrogen ring. So it's 2 rings fused together. Okay, and adenine and guanine are those. And then the pyrimidines are just a single 6-carbon and nitrogen ring. Okay, because they have lots of hydrogens bonded to their nitrogens, they can provide hydrogen to the solution that they're in. So they're bases. So we call them nitrogenous bases. Okay, and you can see there are differences in functional groups between the 2. Okay, so here's a methyl group on thymine where there's no methyl group on these others. This one has an amino group where these 2 don't have amino groups. And so you can sort of pick out different functional groups that give different reactivity to those nitrogenous bases. Okay, so those are sort of all the parts that go into making a nucleotide. All right, so this is how this works. Okay, the nitrogenous bases can very easily form hydrogen bonds with each other. Okay, and when the nitrogenous bases form hydrogen bonds with each other, they're quite particular about which base they can form bonds with. Okay, so what happens is there will always be a purine bonded to a pyrimidine, okay? And it's not any purine bonded to any pyrimidine. It's quite specifically guanine bonding to cytosine, okay? Guanine is the purine, cytosine is the pyrimidine, and adenine bonding to thymine, okay? Again, adenine is the purine, thymine is the pyrimidine. They're bonded together by hydrogen bonds, and you can see here by the little dashed lines, there are 3 hydrogen bonds bonding guanine and cytosine, and 2 hydrogen bonds bonding adenine and thymine. You don't need to have that memorized for the IB. You just need to know that guanine and cytosine will bond together and adenine and thymine will bond together. But it's interesting information to know. Okay? In DNA, this is how bonding occurs with the nitrogenous bases. In RNA, there is no thymine. Okay? So in RNA, thymine is replaced with uracil. And we'll go over I'll show you some pictures of that a little bit later. But just remember, wherever you would see a thymine nitrogenous base, in RNA it's replaced with a uracil. Okay, so let's look at just one single strand here of a nucleic acid. Okay, to orient you, here we have deoxyribose. Oh, yeah, here we have deoxyribose and our phosphate group. Okay, and here's a purine. Deoxyribose, phosphate group, pyrimidine. Deoxyribose, phosphate group, purine. You get the idea. Okay. Now remember, we talked about how to count the carbons in a carbon ring. You go from the right of the oxygen in the ring, so this is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. The way that the nucleic acids are oriented, you'll notice carbon 5 is pointing up, we call that the 5' group, and carbon 3 is pointing down, we call that the 3' group. Okay? So that gives you a basic orientation to a single strand of DNA. Now let's take a look at the two strands together. Okay? They call this orientation or conformation an anti-parallel conformation. Parallel because the backbones are parallel to each other. You can see it here. We go sugar, phosphate, and they're nitrogenous bases. Anti-parallel because notice how on one side the 5' end of the deoxyribose is pointed up, and then on the other side the 5' group of deoxyribose is pointed down. Okay? They're oriented this way to increase their stability. Okay? If it was flipped in the other direction, it would change the conformation and it wouldn't be such a stable molecule. Okay? So the hydrogen bonds between the nitrogenous bases are fairly weak bonds, and the covalent bonds that bind the phosphate groups to the deoxyribose are they're strong bonds. Okay? So it's quite easy to break along this line, and then harder to break the molecule along any one of these lines. Actually, specialized enzymes are required in order to do that. Okay? So water solubility helps to preserve the structure of this molecule, which is important because inside cells, most of the solutions are aqueous, or water-based solutions. It's quite easy to reproduce because you already have a template with any one strand. Because of the fact that adenine always binds with thymine and guanine always binds with cytosine, if there were to be some break here, it would be easy to reassemble because only one type of nucleotide would be able to bond there. This also makes it easy for the cell to proofread because if the wrong molecule binds, it'll actually cause a bulge or a dip, and the actual shape of the DNA will be incorrect. So cells actually have the capability of proofreading. Okay, so the basic things you need to know about the structure of DNA are this. You need to know the basic structure of a nucleotide. You need to know that there's the phosphate, the sugar, and the nitrogenous base. Okay, you need to know how they're oriented in relation to each other, so you need to know how the nucleotides polymerize, what they look like when they form a strand. You need to know how the two strands are oriented, that they're 5' on one side and upside down on the other side. And you need to know what properties the phosphate groups confer, that it makes it water soluble, that it makes it a very strong structure. Okay, DNA is quite remarkable because it can stay in its conformation for actually hundreds of thousands of years, keeping its structure the way it is, just because the sugar phosphate backbone will protect the order of the nitrogenous bases on the inside. So now that you know about the structure of DNA, we can take some time and talk about how it replicates itself, how it organizes itself, and then how the order of the nitrogenous bases actually gives the code or the blueprint for creating proteins, and how those are responsible for all of the inherited information that you have.